Previously, we have talked about different databases, and now when we submit a sequence to a database, it come up with some conflict, like it will say your sequence is not right, or we when check a sequence over there in the database, so the sequence has some specific tags like sequence conflicts. So if these discrepancies are severe, the underlying cross reference to the MBEL is tag, like 4 tags are normally used. So these databases, they use 4 different tags for problematic sequences, and the first of that is frame shift. So so the discrepancies are due to insertion or deletion of 1 or more nucleotide in the sequence. So if there is some insertion or deletion in a sequence that make that sequence frame shift, so that will tag that sequence as a frame shift. So the tag with that sequence will be a frame shift. So when you see a sequence tagged with a frame shift, that will mean there is some insertion or deletion in that sequence which the author needs to modify that sequence. So you have to take care of these sort of sequences. They should not be relied upon. And different so there is another tag, and that will be named as different initiation, like discrepancies are due to an erroneous initiation codon choice in submitted sequence. Like if there is a sequence and that sequence is here, like ATGCT, so like it should be CATG TG. It's like this is a sequence, and somebody starts this like this one, but in original the sequence is like this. ATG is the start codon. Like in original it should be like ATG and then C and then ATG and then TGG and then TG. So in original, like if this is this stop codon, this should be like G. So this stop codon is the actual start codon. Like if this one is the start codon and somebody erroneously incorrectly like started with this codon, so the other ATG is not now ATG, it's not ATG now. This is CAT, and this is so erroneously if somebody like make it like this, so it will say different initiation. समझ आ रही है? You got my point?
Yes, sir.
Okay, and different termination. So the termination codon of the submitted sequence differ from that of the sequence displayed. So somebody like may have like terminated the sequence earlier than the like the stop codon, or like skip the stop codon and go beyond the stop codon and terminate it at different position. So that will be different termination. So the tag with that sequence will be different termination. And next is sequence problem. If there are discrepancies and those are due to erroneous gene model prediction, erroneous open reading frame. So some open reading frame has been detected like that is with errors and miscellaneous some other discrepancies. So the tag will be a sequence problem. So these 4 tags are normally used by these databases with having sequence problems. Can somebody explain this slide so that it is easy for me to like, please? Who will explain? Kalimullah, Kalim, you there? Kaleem, are you
Yes, sir.
there? G, please, if you can explain the slide in your own words.
सर मैं कर दूं।
जी शफीक, प्लीज आप कर दें।
सर जब कोई भी वर्कर जो है या कोई रिसर्चर अगर सीक्वेंस स्टार्ट करता है और उसको चेक करने के लिए ये जो डिफरेंट ईएमबीएल है, जेन बैंक है, इस पे क्रॉस रेफरेंस के लिए चेक करता है, वहां पे सबमिट करता है, तो अगर चांसेस है कि उसने गलती से कहीं जो सीक्वेंस थी उसमें गलत मार्किंग कर दी हो स्टार्ट कोडॉन की या टर्मिनेशन पॉइंट की, जिसकी वजह से डिफरेंट टैग अपीयर हो जाएंगे उसके सीक्वेंस के ऊपर। अगर फ्रेम शिफ्ट अपीयर हो जाता है उसका मतलब है उसने जो ओरिजिनल सीक्वेंस है उसकी जगह कहीं पे या इंसर्शन कर दी एक न्यूक्लियोटाइड की या डिलीशन कर दी है, जिसकी वजह से इंटायर जो है वो जो सीक्वेंस है वो शिफ्ट हो गया। और डिफरेंट इनिशिएशन में जिस तरह आपने बताया कि अगर एटीजी एंड देन एक कोई न्यूक्लियोटाइड कोई भी आ सकता है, उसके बाद एटीजी आता है और हमारा स्टार्ट कोडॉन सेकंड वाले से होना था, तो अगर वो गलती से फर्स्ट वाले कोडॉन को एज अ स्टार्ट कोडॉन मार्क कर देता तो वो वहां पे डिफरेंट इनिशिएशन का टैग आ जाएगा। एंड इसी तरह टर्मिनेशन का भी सेम था कि अगर वो वहां पे जो टर्मिनेशन को बिफोर अपनी तरफ से पहले ही कोई टर्मिनेशन को डोन बना देता है ओरिजिनल वाले से, तो वहां पे जो डिफरेंट टर्मिनेशन का टैग आ जाएगा।
And sequence problems?
सर सीक्वेंस प्रॉब्लम में सर ये नहीं मुझे याद है।
Okay. G, who will explain? Like some other body? ठीक है शफीक, that's good. Fareen is there, Fareen?
Yes, sir.
जी please.
इसमें आपने बताया था कि सीक्वेंस में कोई डिस्क्रिपेंसीज आ जाए तो फिर ये वाले टैग्स लग जाएंगे।
Like किस तरह? Like ये ऊपर जो तीन दिए हुए हैं, other than these three, अगर कोई और discrepancy आ
Yes.
जाए, like we need to describe, we need to annotate an open reading frame. We need to describe an open reading frame that is present from nucleotide number 10 to nucleotide number 100. And due to some error, we say that nucleotide number 15 to nucleotide number 100. So that is like we are predicting some other open reading frame now. So
Yes,
like
sir.
okay, so
Formers.
so in that case there will be like the tag will be sequence problem and not like frame shift, different initiation or different termination. That will be not that case actually. And some other miscellaneous discrepancies can be there, like okay, we will discuss these in detail in later slides as well. So uh so to the whole class, am I clear?
Yes, sir.
Okay. And Faiza Kudus, she asked that what is canonical sequence? Canonical sequence is the term is actually used for most accepted like concept. Like canonical means the thing which is more accepted and the common pathway and the common thing. Like we know a sequence start with like some specific start codon and that start ends with some another. Like we know ATG is the start codon and TGA, TAC, TAG, TAA, they are stop codons. So this is canonical. But in mitochondria, the start codon is not ATG. That could be some like other sequence. So the canonical, so mitochondria is the start codon is not canonical in mitochondria. So a canonical is a general term and that can be applied for the most accepted thing. Yeah, accepted like problem or accepted procedure or accepted cycle, anything which could be there. So next is cross references to the sequence databases. The discrepancies are actually described in more details in sequence caution. So there will be a tag, there will be a caution or a caution with a sequence caution. And if no coding sequence is there, means no CDS has been annotated. If you have submitted a sequence, if I have submitted a sequence and I didn't annotate the sequence, annotation means I didn't tell about my sequence, that where the start codon is there and from where there will be the termination sequence is there, or the promoter is there. I didn't annotate anything. But if I didn't say about coding sequence, so if no coding sequence has been annotated in the submitted sequence, most frequently genomic sequence, the corresponding cross-reference is tagged with the label no translation available. So if we don't annotate our sequence, if we don't say about our sequence, like we don't say, like we don't locate the start codon and the stop codons and the translation product, if we don't locate them, the data bank will annotate our will have this caution, and it will be like sequence caution, and the label will be no translation available. So that will mean the author did not provide any information about the coding sequence. And there is an example for that. If you put this accession number in the GenBank, it will provide you the sequence, the problem. And if you can see this thing, sorry, that was Uniprot. Okay, so if you can see here, they are with caution, like genomic DNA translation. You can see frame shift. So that will, these are the labels, and you can see the sequence. This differ from that shown reason frame shift at position 20. So reason frame shift at position 20. So this is how the sequences are, the databases, they put some notes with the sequence if there are problems. And next is there are two types of molecular databases. The first one is primary databases, and the second one is derivative databases. The primary databases, they are the original submissions by the experimentalist. They are original submissions by the scientist, and content controlled by submitter. The person who submit that, they can control the content of that submission. Like if you submit a sequence to the database, you can edit that, you can control the sequence, the sequence information, for example, GenBank, etc. So when we need to submit a sequence, so we will submit into the primary databases, and we can like edit that sequence. Similarly, for derivative databases, they are actually derived from the primary data, and we will not control them. The databases will control them, and the whole responsibility lies on the databases, the authorities of the databases. So derived from the primary data, controlled by third party, the NCBI. So NCBI normally controls the secondary databases or derivative databases, and examples are NCBI protein. So the NCBI protein, ref sequence, ref SNP. So if you can see, the NCBI, the NCBI proteins, they are actually the proteins that have been derived from these sequences. We submit a nucleotide sequence and they derive protein from them. And they have another the derivative database for that. Reference sequence, they select a sequence and annotate them and call that as a reference sequence. This is just like a reference book. We have different book on the same subject, but the library or some authority, they will identify one of those specialized book as reference book. So when you go, or when we go into a library, we can observe a different section known as reference section, and in that reference section there are books known as like reference books, and they are reference book on some specific topics. So and that will mean they are authentic books. Similarly, these reference sequences will be authentic sequences, because we cannot rely more, or we cannot rely at more extent on these sequences, because they may have containing some errors. But these reference sequences, they are authentic. Like if you want to work on coronavirus, so just go on these databases, and if you want to have some authentic sequence of that, so go for reference sequence of coronavirus. Don't go for other sequence, because the other sequence may have some errors. So go and download reference sequence from there. So reference sequence will be the authentic one. And similarly, reference SNPs. So we will be discussing about SNPs later on, but reference SNPs are also there. If you want to have reference SNPs, so you can get that. So this is SNPs, means single nucleotide polymorphism. You may have studied this like in the other subjects as well. So and that's it. इसकी समझ आ गई है पूरी क्लास को? Am I clear to
Yes,
all
sir.
of you?
सर ये जो प्राइमरी
Yes, sir.
डेटाबेस में है कंट्रोल सबमिटर वाला ये समझ
Okay.
But
So that will mean that when, like Kalim, if you have a sample of blood, or like DNA, you do PCR, and then you go for a sequencing. You have a sequence, and now you need to submit that sequence to the database for other people to check. So what will happen? You will submit the sequence, you will submit the information there, and at some another point, another time, like 10 days after, 1 week after, or a year after, you can make changes over there. And the although the database will like generate another like GI number and so on, we have already discussed those things, but you have control over this sequence, this data. Like you can change information in the sequence. But in derivative sequence, derivative databases, these databases, they are derived from the primary databases. We don't have control on these. They drag information from these. So the whole panel of the people present in for these databases, they will control the data. They will drag information from these sequences, and that will be like that they will be like derivative databases. We can't make changes in these databases. We can't make changes in reference sequences. We can't change in NCBI proteins like these. So we can't change them. But we can change our own sequences, like when we submit there here in these databases actually.
सर
Am I
प्राइमरी
clear?
डेटाबेस सर प्राइमरी डेटाबेस में ऐसे जैसे कि हम कोई डाटा सबमिट करते हैं लेकिन प्राइवेट में हम कहाँ से ले कहीं से लेके फिर उस आगे सबमिट करते हैं
हाँ, डिराइवेटिव डेटाबेसेस, एग्जैक्टली। डिराइवेटिव डेटाबेसेस जो है वो इन से डेटा लेते हैं। वो लोग जो इन डेटाबेसेस को चला रहे हैं, वो इन्हीं सीक्वेंसेस से डेटा लेते हैं, प्राइमरी डेटाबेसेस से, और
यस
डिराइवेटिव डेटाबेसेस में वो डेटा जनरेट होता जाता है, ऐड होता जाता है वो। जी जी, एग्जैक्टली। जी, अच्छा। एंड व्हाट नेक्स्ट? इसकी समझ आ गई है पूरी क्लास को?
Yes.
Yes,
मजीद, आपको समझ आ गई है?
yes, sir.
ओके, ठीक है। Okay, and next is okay. So the difference between Exom, Intron, CDS, ORF, UTR. So here you can like understand what's the difference between all these terms. Like what is Exon, what is Intron, what is CDS, what is open reading frame, what is UTR. So this is a sequence of DNA. and this is the 3' region. This is the 5' region. The color in blue that contains transcription factor binding sites, TOTA box, CCAAT box. So, and this is transcription start site. So this is transcription start site, not translation. Here, this is Exon, this is Intron, this will be Exon 2, this will be Intron 2, this will be Exon 3, and this is 3' region. When transcription occurs, it will produce RNA. And now the RNA is from here to here. And that is the regulatory region in the DNA for this RNA, for this region actually. Now this transcription when occurs, it will produce like RNA from this region to this region. And now the polymerase has to bind in this region, and they will start from here. Like but the binding region will be here in the promoter region. And now when the RNA is formed, the primary transcript is formed. This is the downstream element, and this is exon 1, intron 1, exon 2, intron 2, exon 3, intron, and so on. Okay, so what will happen from the exon 1? So after splicing, what will happen? When splicing occurs, there are regions that like GT, AG, GT, AG, and so on. So these introns will be spliced out. The orange region, they will be spliced out, and the green region, they will combine to make a mature messenger RNA. Poly-A tail will be there, and this is the cleavage site, and poly-A tail will bind there, and it will be polyadenylate, like polyadenyl adenyl transferase will add poly-A tail there, and mature RNA will be produced. So now in the mature RNA, there will be capping. So we already have studied about capping, how capping will occur, how poly-A tail will bind, will be produced, and the cleavage site is here, and the introns, how the introns are spliced out. So now the RNA is mature. In this mature RNA, so you can see the cap region is there, and in this exon, the exon can be divided into two parts. Now this is 5' UTR, the 5' untranslated region. So the exon doesn't mean the whole of the exon will produce protein. It has untranslated region as well. The exon has untranslated region. So it is the part of the exon containing untranslated region. So the untranslated region means the ribosome can bind there at this untranslated region, and this start codon is here. So AUG will be here. AUG is here. So this is the start codon from here. So now the start codon is here, and after that the second exon is there, and after that the third exon, part of the third exon is there, and this is now, this will be spliced out. This will be like this is the stop codon here, and this is 3' untranslated region. So this will be, and here the poly A signal is there. So this is 3' untranslated region from here to here. So this will not translate. So start codon is here, that is the part of the exon, and stop codon is here, that is also the part of the exon. 3', 5' UTR is here, that is the part of the exon, and 3' UTR is there, so that is also the part of the exon actually. So now the cleavage site within the exon is here, and after cleavage here, that will cleave out, and the poly-A tail will bind there, and it will produce the final mature messenger RNA. So within this mature messenger RNA, which protein will form, like how, from which region to which region the protein will form, from the start codon to the stop codon. So that lies within the exonic within the primary transcript. So to deleting the 3' UTR and 5' UTR, the rest one from the start codon to the stop codon, that will be called as CDS, coding sequence. So the coding sequence means starting from the start codon to the stop codon. That is the coding sequence. Coding sequence doesn't mean, don't confuse it like with exons. The coding sequence is the part of the exon. Exon also contain untranslated region as well. So this is only the translation region. The coding sequence is the translation region. So after translation, the protein will form from here to here only. Is it clear?
Yes.
And this
Yes,
is the
sir.
another so this is another representation of this. This is the cap region, the 5' UTR start and stop. So this is the coding sequence, and then 3' UTR, and that is then poly A tail. G please.
सर ये सेकंड स्टेप बता दे जब आपने बाइनरी वाला पॉइंट बताया था, ये डाउनस्ट्रीम एलिमेंट वाला।
ये पॉइंट
सर जब वो आरएनए बन गया था, ये आपने बताया था ना यहाँ पे बाइनरी वाला पॉइंट बताया था ट्रांसक्रिप्शन के बाद। जी सर यही वाला।
हाँ, ट्रांसक्रिप्शन के बाद क्या होगा कि यहाँ पे 3' UTR में कुछ रीजन होगा जो अनट्रांसलेटेड रीजन होगा, 3' UTR होगा, 3' अनट्रांसलेटेड रीजन, इसने ट्रांसलेट नहीं होना। स्टॉप कोडॉन विल बी हियर, सो फ्रॉम हियर टू हियर दैट विल बी कोडिंग सीक्वेंस। ये जो है अब इस अनट्रांसलेटेड रीजन में ये 3' एक्सॉन 3 में क्या होगा, देयर विल बी अ पोली ए सिग्नल। The poly A signal is actually the cleavage site. यहाँ पे cleavage होगी, cleavage होके फिर poly A tail बनेगा, means के poly A tail will bind there. So poly adenyl transferase जो है वो यहाँ पे poly A tail add कर देगा. So this mature messenger RNA will be produced, and the translation will then start from here to here, and the ribosome will bind there. ठीक है? Am I clear?
यस सर।
कि कौन एक्सप्लेन करेगा आप इसको, इस पूरे डायग्राम को? Who will explain? Please come on. Please.
सर मैं बताऊं?
जी फरीन आप बता दो जी। कलीमुल्लाह इसके बाद आपने बताना।
तो इसमें आपने बताया कि ये डीएनए का हमारे पास स्ट्रैंड है 5' to 3', जो लेफ्ट साइड पे प्रोमोटेड रीजन है, यहाँ पे ट्रांसक्रिप्शन बाइंडिंग फैक्टर्स हैं, और इसके साथ एक्सॉन 1, इंट्रॉन 1, एक्सॉन 2, और इस तरीके से। अब जब ट्रांसक्रिप्शन होगी सर, तो फिर वो जो हमारे पास रीजन आ जाएगा वो एक्सॉन 1 से इधर डाउन स्ट्रीम तक आ जाएगा। तो फिर सर आपने बताया फिर दोबारा स्प्लाइसिंग होगी तो इंट्रॉन्स जो होंगे उनको स्प्लाइस आउट कर देंगे, और जो एक्सॉन 1 है वो भी स्प्लाइस होगा, और उधर से एक्सॉन 3 भी आधा हो जाएगा। जो इधर वाला है उसके अंदर यूटीआर कैप के सिग्नल्स होंगे, और जो राइट साइड पे उसके पॉली ए सिग्नल्स होंगे उसकी टेल के। तो फिर सर इस तरीके से जब स्प्लाइस आउट हो जाएगा तो इधर टेल आ जाएगी, उधर 5 कैप आ जाएगा। और फिर आपने बताया कि सीडीएस जो होता है वो ट्रांसलेशन रीजन होता है फ्रॉम स्टार्ट कोडॉन टू स्टॉप कोडॉन। तो फिर सर वो उधर से दोबारा ट्रांसलेशन होगी तो हमारे पास प्रोटीन आ जाएगी।
एग्जैक्टली। कलीम कोई इस तरह कोई इसमें
यस
बात
सर।
रह गई हो तो आप ऐड कर दें इसमें।
सर इसमें ऐसे जैसे आपने बताया कि पहले पहले प्रमोटर लगा तो ट्रांसक्रिप्शन हुई है, ट्रांसक्रिप्शन के बाद सप्लाइसिंग, सप्लाइसिंग से जैसे फर्स्ट कुछ पार्ट है जैसे इधर एक्सॉन 1 में तो स्टार्ट कोडॉन आगे लगा, जो पहले वाला जो पार्ट था वो एस 5 यूटीआर बन अनट्रांसलेटेड रीजन बन गया, और उस साइड भी ऐसे अनट्रांसलेटेड रीजन बन गया, और जो एंट्रॉन पार्ट थे जो वो सप्लाई आउट हो गए। उसके बाद ट्रांसलेशन हुई, जो समझो अनट्रांसलेटेड पार्ट उनकी उनकी ट्रांसलेशन नहीं हुई, और प्रोटीन लास्ट बन गई।
ओके, दैट्स गुड। किंजा इज देयर? किंजा माजिद। खंसा शहजादी, खंसा, don't be dormant please, come on. लाइबा इरफान।
जी सर।
जी आपको समझ आ गई है?
जी सर आ गई है।
लाइक किस चीज की, लाइक कोई पॉइंट अगर रह गया हो तो वो
सर
बता दें प्लीज।
इंट्रॉन्स के बारे में आपने इतना नहीं बताया, बस यही बताया था कि वो जब स्प्लाइसिंग होती है तो वो रिमूव हो जाते हैं, उसके बारे में थोड़ा सा बता दें।
अच्छा ओके वेट, Okay, Kinza Majid, your mic has problem. Okay, no worries. Okay, so introns के बारे में ये था, like I didn't go in detail into the story of exon and introns and how the splicing occurs and how the transcription occur and how the translation occur. I don't go into the detail, and the reason is you have already subject, like you already studied molecular biology courses like molecular biology 1 and 2, and they are meant for the details of these, but that is another aspect. We are actually covering bioinformatics. But for as you asked, the introns, they are actually the spliced out region. The spliced out, they are not the part of the mature messenger RNA, and that region is known as actually intron. And same is if that the same region occurs in the proteins, and the proteins, the splice out region in the protein will be called as intins, and the spliced out okay, and the remaining region will be known as exons, and that will be in the protein. But in the DNA, sorry, in the RNA, the spliced out region, spliceosome will splice out, will remove this region from the RNA. Actually, how we got to know, like why, which region is removed out, so and is spliced out. So we will have a sequence of the DNA. Also, we have a sequence of the like the protein product. So by comparing the protein product with the DNA sequence, we can know which region has been spliced out.
सर
जी Noreen, please.
सर ये ओपन रीडिंग फ्रेगमेंट और ये कोडिंग सीक्वेंस में इन दोनों में क्या डिफरेंस होगा?
ओपन रीडिंग फ्रेम कहाँ पे है?
सर ये ऊपर लिखा हुआ है ना, हम लोगों ने मॉलिक्यूलर में पढ़ा था कि जो रीजन ट्रांसलेट हो रहा होता है उसे हम लोग ओपन रीडिंग फ्रेगमेंट कहते हैं। तो
हाँ,
इस यहाँ पे
so
क्या डिफरेंस होगा
in the coming
कोडिंग सीक्वेंस में?
slides, okay, in the coming slide we have a detailed comparison of these two things. They are actually different things. They are not the same. But
जी सर।
for the coding sequence for now, actually things got confused. Therefore, I didn't add operating frame here. In the coming slide, we will compare CDS and operating frame. So for now, we got to know Exon, we got to know Intron, we got to locate CDS, we got to locate UTRs, 5' and 3' UTRs. Am I clear?
यस सर, बाकी सब समझ आ
Okay.
गया।
Okay, and that's nice. And if you can see here, the TSS is transcription start site, and the AG here, AG, and GT. So they are the spliceosome recognizes these two sequences, and on the exon intron junction, they are at the intron exon junction, and they splice out the intron, they splice out the introns by recognizing these sequences. Am I clear? समझ आ रही है?
Yes, sir.
Okay, that's good. Okay. And next is open reading frame. So now the story starts with the open reading frame. What's the difference between open reading frame and the CDS? CDS or open reading frame में फर्क क्या होगा? क्योंकि usually we confuse both terms. So अभी इसको समझ लें. The region of the nucleotide sequences from the start codon to the stop codon is called open reading frame. Start codon से stop codon तक जितना भी region होगा उसको हम क्या कहेंगे? Open reading frame कहेंगे. ठीक है? And depending on the start point, there are 6 possible ways, 3 forward and 3 on the complementary strand of the translating any nucleotide sequence into amino acid sequence according to the genetic code. These are called reading frames. So अगर अभी भी clear नहीं है, so I will make it more clear for you. I will try to make it more clear. Like if that is a sequence, like if we have a sequence CT, CGT, इस तरह का अगर sequence है, if this is a sequence, so we have one sequence, this one, so we will call it 0 frame. So this is a 0 reading frame. This one is 0 reading frame, means start from here. This will be GTC and GTG and ATA and GAT. So this will be, we will refer this as a 0 reading frame. So if I can make it -1 reading frame, or if I call it +1 reading frame. So what will be +1 reading frame? That will be like +1 एक nucleotide आगे जाके, like I will skip one nucleotide and will start from here TCG, फिर TGA and TAG and AT. So now you can see this start from here. If I delete this, so I start from here. So this will be +1 reading frame. Means translation यहाँ से शुरू हो रही, यहाँ पे translation यहाँ से शुरू हो रही थी ना, तो राइबोसोम ने क्या करना है, एक न्यूक्लियोटाइड को एक न्यूक्लियोटाइड स्किप करके आगे जाके शुरू करेगा. So that will be +1 reading frame. And we also have -1 reading frame. We also have -1 reading frame, and that will be -1. What will be -1? So if we have a sequence here like A, so if we have a sequence here A, like if we have a sequence here, okay, and that's it. Okay, so G T C G T G A T A G A and then T. So if we delete this one now, so sorry, Istra. So this will be, so this is 0 frame, this is +1 frame, and this is -1. So what will happen? Actually, the ribosome start from here. We consider this as 0 frame. So in this case, when the ribosome come, it will start from here. It will skip this one nucleotide and start from here and making it this sequence, this codon, and this codon, and this codon. And with this codon, it will bind, it will add amino acid according to this codon. Now, if that is -1, in -1 case, the ribosome will bind and will go back one nucleotide from here, and it will like AGT and then CGT and then GAT and so on. So actually now there are 3 operating frames, 3 possible operating frames from one sequence. From a single sequence, there are 3 possible open reading frames. So in DNA, we have 2 strands. In DNA, we have 2 strands. And so it could happen that the ribosome scan the sequence from this side, and also the ribosome can scan from this, because this will be 5 to 3 and this will be 5 to 3. This will be 5 prime end, and this will be also be 5 prime end. So, and the sequence will be the same because this is complementary to this sequence. So, and the sequence, like if we can draw, so the ribosome will scan in this direction. Here, the ribosome will scan in this direction. So 3 possible reading frames are possible from this sequence, 3 possible from this strand, and 3 possible from this strand. So we have by rule 6 possible ways to, we have 6 possible ways, 3 forward strand and 3 complementary strand for translating any nucleotide sequence into amino acid according to the genetic code. Am I clear?
Yes, sir.
So all these 6 reading frames will be called as open reading frames. समझ आ रही है? And all these will start with ATG and all these will end with the stop codons, with the one of the 3 stop codons. Am I clear?
Yes, sir. इसी के साथ अगर आप CDS भी बता दें सर.
Exactly. I'm going towards that. I'm trying to go towards that. Okay, and all these 6 are possible, but the actual, which is like actually translating product in the living organism, yeah, or in vivo translation, the actual region, the actual reading frame which the ribosome select is the CDS. Means हमारे पास 6 open reading frames हैं, पॉसिबिलिटीज 6 की हैं, लेकिन राइबोसोम किसको सेलेक्ट कर रहा है? लाइक राइबोसोम सिर्फ इस रीडिंग फ्रेम को बना रहा है, इस रीडिंग फ्रेम को ट्रांसलेट कर रहा है, या इस रीडिंग फ्रेम को कंसीडर कर रहा है, बाकी 5 को कंसीडर नहीं कर रहा. सो वो जो सेलेक्टेड रीडिंग फ्रेम है, दैट विल बी कॉल्ड एज CDS, coding sequence. That will be called as coding sequence. The rest of the 6, all of the 6 will be called as open reading frames, but the actual reading frame which is selected by the ribosome will be called as coding sequence or CDS. Am I clear?
Yes, sir.
That's good.
Yes,
That's good.
sir.
सर
That's nice. जी please.
सर ये कॉम्प्लीमेंट्री स्टैंड की समझ नहीं आई. ट्रांसलेशन में तो मैसेंजर आरएनए का सिर्फ सिंगल स्टैंड ही होगा 5' to 3'.
हाँ, exactly. इससे पहले देखें हमारे पास अगर if we have a sequence, so the polymerase can work on this to make an RNA. The polymerase also can work on this to make an RNA.
यस
समझ
सर.
आ रही है? ठीक है.
यस
तो
सर.
हमारे पास दो टाइप के RNA पॉसिबल हैं. सो एक ही
ओके सर.
DNA सीक्वेंस से हमारे पास 2 RNA पॉसिबल हैं, और मींस 6 possible ways to translation is possible.
ओके सर.
ठीक है. That's good. That's good. Am I clear to the whole class?
Yes, sir.
Yes, sir.
That's nice. That's nice. And going further, so CDS now, I assume you already understand this thing. The actual region of the DNA that is translated to form protein. So this will be the actual region. They are the possible regions, the possible translation products, but this is the actual translation product. The CDS refers to those nucleotides, concatenated exons that can be divided into codons, which are actual translation into amino acid by ribosomal translation machinery. So from this, so this will be the actual. So this, so the ribosomal machinery actually, the ribosomal machinery actually selects a sequence, and that sequence will be called as CDS. And next is operating frame versus CDS. CDS means only the sequence that is known to be transcribed, and therefore the coding for something. Any full messenger RNA sequence obtained from the cDNA sequence will have the full coding sequence. So, so we already have discussed this thing. So operating frame is usually predicted based on DNA sequence and not proven to be transcribed. So operating frame जरूरी नहीं है कि वो transcribe हो सके, ठीक है? And they are not proven to be transcribed. But the sequence, the CDS is proven to be transcribed. Actually, they are proved translation products. Am I clear? Okay, let me check the chat. Okay, Faida Kudus, the Faiza, the concatenated sequences and the concatenated exons, these are concatenated exons, 1, 2, 3. So they are concatenated means they can be grouped together. Am I clear? Multiple sequences and they are grouped together. Good. And next is, so now we need to understand the difference between a sequence and a string. So a sequence is an ordered list of elements, usually infinite. So the sequence of positive integers, so this is infinite sequence. So the sequence which we refer to a sequence is not a sequence. Sequence by definition is what? Is infinite. So it should not stop like somewhere. So a sequence, and it should have some sequence, some order. So a sequence is an ordered list of elements. So it must have some order, and therefore we will call it sequence. So like sequence of positive integers, and that will mean the order is there, like 1, 2, 3, 4, 5. You can assume, like you can predict the sequence like 1, 2, 3, and after that there must be 4, 5, 6, and so on, and there that should be infinite. It will not stop. That will not be finite. That is infinite. And a sequence of Fibonacci numbers, the each one in the sum of two previous number. So Fibonacci number, what are they? If you can sum up two number, the third will be there. So so the sequence of Fibonacci number is will be 0 plus 1 will be 1, 1 plus 2 will be 3, 3 plus uh, so uh, 2 plus 3 will be 5, and 3 plus 5 will be 8, 5 plus 8 will be 13, and you can predict the of others. So here you can see 0 plus 1 is 1, and 1 plus 1 is 2, and 1 plus 2 is 3, and so on. So you can predict the numbers. So there is a sequence there. There is a sequence in there, in there, and also this is an infinite sequence. So that is by definition a sequence. Similarly, if we remove elements from a sequence, we obtain a subsequence. For example, we remove all the odd number from the sequence of positive integers. So if we remove the odd numbers, so that will be a subsequence. We get the subsequence consisting of even positive integers. So the even positive integer, so this is now a subsequence that has been removed, that has been dragged from positive integers. So now they are even positive integers. So even positive integers, but again the subsequence also will be like infinite. And what is now string? So string by definition, if given a set of x, a string over x is a finite order list of elements of x. So see, if x is the set x, like a, b, c, then following are the numbers of string and the example of strings over x. So if this is a set, like ABC is a set x, so what is a string over x? So that could be, a string could be the ABA, a string could be double a string could be double BA, and so on. So they are derived from this set. So this will be a string over x. So a string over x could be any of the. So if you assume a sequence, what we assume like a sequence of nucleotide. So actually that is actually not a sequence, that is a string. Like we have 4 nucleotides, and if you consider that a set of 4 nucleotides like ATGC. So nucleotides could be a DNA, could be a string over x, and that could be like if we can observe a DNA, So that is a string over X actually. So A, T, G, T, G, C, and so on. So that could be like a string over X. So whole of the nucleotide sequence, what we call as a sequence, is actually a string. समझ आ रही है कि नहीं?
सर ये हमने किस तरह इनको बनाया है, मतलब एक दफा ए आगे वो 4 टाइम्स ए आ रहा है, फिर ए बी आ रहा है, ये किस तरह हमने ड्राइव किया, ये थोड़ा एक्सप्लेन कर दें एग्जांपल से।
जी, असल में इसमें ये बताया गया है कि हमारे पास एक सेट है वो ए बी सी का। लाइक मैं इसको एक और एग्जांपल से बता दूं। अगर मेरे पास एक सेट है x एंड x इज इक्वल टू ए टी जी, सॉरी इसको मैं स्मॉल लिख देता हूं, ए टी जी सी। सो व्हाट विल बी अ स्ट्रिंग ओवर x? लाइक मेरे पास एक डीएनए सीक्वेंस है। सो डीएनए सीक्वेंस जिसको हम डीएनए सीक्वेंस कहें, लेकिन वो एक्चुअली स्ट्रिंग है, तो वो क्या होगा? ए टी, सॉरी, ए टी सी जी टी जी। ये एक है, तो व्हाट इज दिस एक्चुअली? सो कैन यू सी, कैन वी कॉल इट अ सीक्वेंस? हम इसको सीक्वेंस कह सकते हैं, हम तो सीक्वेंस कह रहे होते हैं ना, बट इन एक्चुअल बाय डेफिनेशन दैट इज नॉट अ सीक्वेंस। A sequence means what a sequence means. A sequence means infinite. This is not infinite. A sequence means order, some specific order, like integers, like Fibonacci numbers, like odd integers, like something like, so there must be some order, but this doesn't have some order. So by definition, what the sequence DNA sequence is, by definition, it is string actually. लाइक ये नंबर जितनी दफा भी इस सेट से कोई भी इंटीजर, कोई भी डिजिट अगर हम ले लें, जितनी दफा भी रिपीट कर लें, तो वो इसका सबसेट होगा, सब स्ट्रिंग होगा। ठीक है, सो स्ट्रिंग ओवर एक्स विल बी, सॉरी, तो सो वो जो है दैट विल बी अ स्ट्रिंग और दैट सेट। सो उस सेट से डिजिट्स लिए गए हैं और वो रिपीट हो रहे हैं बार बार। ठीक है, सिमिलरली इस सेट से ये डिजिट्स लिए गए हैं और वो रिपीट हो रहे हैं, तो ये एक्चुअली जिसको हम सीक्वेंस कह रहे हैं, दैट इज एक्चुअली अ स्ट्रिंग। एम आई क्लियर?
यस सर।
यस सर।
अच्छा, कौन इसको एक्सप्लेन करेगा? अपने वर्ड्स में एक्सप्लेन करें, शायद मुझे ज्यादा एक्सप्लेन करना नहीं आ रहा इस तरह। जी,
इन दोनों में डिफरेंस मेजरली यही डिफरेंस आएगा ना कि सीक्वेंस में जो है वो नंबर्स चेंज होते रहते हैं और कोई भी नंबर आ सकता है इनफाइनेटली, लेकिन स्ट्रिंग के अंदर जैसे आपने ये सेट बताया ATGC, तो इसी के अराउंड जो है वो सीक्वेंस स्ट्रिंग की एक बनती जाएगी बस ATGC के अराउंड ही।
यस, एग्जैक्टली एग्जैक्टली, बिल्कुल एग्जैक्टली। सो सो जिस डीएनए सीक्वेंस को हम सीक्वेंस कह रहे होते हैं, वो एक्चुअली क्या है?
स्ट्रिंग।
तो स्ट्रिंग
एक्चुअली
है।
दैट इज एक्चुअली स्ट्रिंग। ठीक है, लेकिन वो इतना लाइक आम हो चुका है कि उसको लाइक हम सीक्वेंस ही कह देते हैं फिर। ठीक है, सो देयर आर सर्टेन टर्म्स नोन एज क्लेशे, कि वो लाइक बहुत कॉमन होती हैं, उसको आप चेंज नहीं कर सकते। लेकिन वी हैव टू अंडरस्टैंड, बीइंग अ बायोइन्फॉर्मेटिशियन, we have to understand the difference, the basic difference between the things. ठीक है, is it clear?
तो ये एक
यस
बार ये जो आपने सम करके बताया सीक्वेंस में, ये वाला दोबारा बता दें, हमने सम क्यों किए हैं सीक्वेंस?
ओके, ये हमने सम नहीं किए, ये बाय डेफिनेशन फिबोनाची नंबर्स की डेफिनेशन में आता है। ठीक है, फिबोनाची नंबर्स कौन से होते हैं? जिसमें हम दो डिजिट्स को ऐड कर दें और वो थर्ड डिजिट बना दें, थर्ड। तो उस तरह के सीक्वेंस में हम जाएंगे, और ये फिबोनाची नंबर्स बाय डेफिनेशन इस तरह है, लाइक 0 और 1 को अगर हम प्लस करें तो इट विल बी 1। अब 1 और 1 को प्लस करें, दैट विल बी 2। 1 और 2 को प्लस कर लें, दैट विल बी 3। and 1 or 2 को plus करें तो that will be 3, 2 or 3 को plus करें that will be 5. So Fibonacci number by definition is like a sequence that is like the sum of 2 previous ones. That each one will be the sum of 2 previous one. So this is the definition of this Fibonacci numbers. Just like odd numbers kya hai, positive integers, why they are positive integers, like and what is the odd number in those integers, and what are the even number in those integers. Am I clear?
Yes, sir.
Okay, and next is biological databases. So the data formats of biological databases is XML. XML जो है ये biological databases का format होता है, XML data format. ठीक है, इसमें लिखा जाता है biological databases को, like they are written in XML and they are understood in XML. There is another format known as HTML. आपने बहुत ज़्यादा सुना होगा HTML. So these are the
Yes,
two
sir.
formats, HTML and HTML. So XML and HTML are two different formats. So what XML is? So XML is extensible markup language, XML, and XML is a markup language much like HTML. ये HTML की तरह है, लेकिन ये जो है उस HTML की तरह markup language है, लेकिन this is extensible and designed to store and transport data. तो इसको किस लिए यूज करते हैं इस लैंग्वेज को? इसको दिस इज एक्चुअली कंप्यूटर लैंग्वेज. इसको यूज इस वजह से करते हैं टू स्टोर एंड ट्रांसपोर्ट डेटा. स्टोर भी कर सकें और उसको ट्रांसपोर्ट भी कर सकें फ्रॉम वन टूल टू अनदर. सो इसको हम ट्रांसपोर्ट कर सकें. सो and set of rules for encoding documents in format that is both human readable and machine readable. हमने इस तरह ये XML is a set of rules and they encoding documents. So the documents we write is they are encoded in such a way that human can read and the machine means the computer can read. So both human and machine could read those encoding documents and those formats actually. And XML has come in common use for interchange of data over internet. So so the data could be interchanged over internet, they could be transported, they could be stored, and XML is the source for that. And the XML is just information wrapped in tags. So the information wrapped in tag is actually the XML. And if you can see this, what are the tags and what are they? Actually, this node, this is an example for that. This node is a node to tow from Johnny. So tow from Johnny stored as XML. So ये information is stored in XML and this is the output for that. So ये information जो है ये XML में किस तरह store होते हैं वो इस तरह आप देख लें कि node से start होगा and it ends on the node. So ये जो slash का निशान है, that will mean so that is the end for this node. So this is the start and this is the end, and this will be bound with these symbols. So this is so this is the tag. This tag is the node. So here you can see the node. This is the output for that, and here it ends. So you will not see the end. So the node is there. So the node is there, and two. So the two is there. Tov. So there will be tov, and two should end. Also, the two should end. So the slash mean two is start and two is end. And here you can see no the end, and from and this will be tag from and Johnny, and from should also end here. So from Johnny, and reading, sorry, heading should also end here. So the heading is reminder. So the reminder is here, and body of the text is body, and body should also end there. So body is don't forget me this weekend. So here is the message, the body of the text. So don't forget me this weekend. and the note is end there, and that's it. इसकी समझ आ गई है?
Yes, sir.
Okay,
Yes, sir.
and next to it is what is the difference between XML and HTML? XML was designed to carry data with a focus on what the data is. Actually, XML has more focus on the data. But HTML means hypertext markup language. XML is extensible markup language. The HTML is hypertext markup language, was designed to display the data with a focus on how the data looks. आपने देखा होगा the web pages, how those looks. The web pages are they are designed in HTML. But the databases, they are designed in XML, because the websites, they are more focused on their aesthetic value, how they looks. But the databases, they are more focused on the data. Therefore, they use XML, and the web pages, they use HTML. And next is XML tags are not predefined like HTML tags are. So, and in XML, the author must define both tags and the document structure. So in XML, the tags are not defined. We have to define the tags first, and then we'll add the data. In HTML, the tags are predefined, already defined tags. We have to take the tags from a list of tags in HTML. But in XML, we have to define the tags. That these tags, we have to define them first. And HTML works with predefined tags like heading 1, table, etc. So they have a list of defined headings and defined tags, and we can use these tags to define our work, to define our what we want to show on a web page, etc. Is it clear?
Yes, sir.
That's good. So extensible markup language, so it contains, many computer system contains data in incompatible formats. So data जो है वो incompatible formats में computer में पाया जाता है, and they have the incompatible formats, so they cannot be transferred from one format to another formats. They cannot be checked in, like आपके पास अगर word नहीं है, तो आप जो है word document read नहीं कर सकते. So that is, आप उसको PDF में read नहीं कर सकते, because the format is incompatible. ठीक है. So exchanging data between incompatible system or upgraded system is time consuming task from web developers. So upgraded system, so फिर क्या होगा कि large amount of data must be converted, and incompatible data must be and is often lost. So incompatible data जो है वो they got lost. So so XML has the beauty of that. XML store data in plain text format. प्लेन टेक्स्ट फॉर्मेट के फॉर्म में एक्सएमएल डेटा को स्टोर करता है. दिस प्रोवाइड अ सॉफ्टवेयर एंड हार्डवेयर इंडिपेंडेंट वे ऑफ स्टोरिंग, ट्रांसपोर्टिंग एंड शेयरिंग डेटा. So the data could be shared without any dependence on a specific software, without any dependence on the specific hardware, and this could be shared in any format for storing, transporting, and this stuff. And XML also makes it easier to expand or upgrade to new operating system, new applications, new browser without losing data. So this is the beauty of the XML. And with XML, data can be available to all kinds of reading machines, like people can read them, computers can read them, voice machines can read them, new feeds, etc. So they can be understood by multiple types of formats, in multiple formats. So for different types of data, the scripts cannot be read by people like in many things, in many formats. So XML is more easy to be understood by the people, by the computer, by the voice machines, and so on. And again, the same thing. Markup refers to sequence of characters or other symbols that you inserted at certain places in a text or word processing files to indicate how the file should look when it is printed and displayed to be described the document's logical structure. The markup indicators are often called tags. So these indicators are often called tags, and this is the printout product. So this is the printout product, but in the machine, it will be stored like this. And practical, this is for you for the practical session. So integrated systems, like you have to go to the NCBI online. So you can go for nucleotide, protein, gene, omim, etc. Omim is actually online Mendelian inheritance of man. So upgraded catalog of human genes, genetic disorder and traits with particular focus on molecular relationship between genetic variation and phenotype expression. So you can go to the integrated system. In this integrated system, multiple softwares have been added on here. NCBI पे आपको different software, different like like GenBank भी मिलेगा वहाँ पे आपको, वहाँ पे PubMed भी मिलेगा, वहाँ पे databases, different databases आपको मिलेंगे. ठीक है? So NCBI is actually an integrated system, and protein databases are there. So you need to go through it, and XPacy is the example for that. And next is protein structure databases are there. PDB sum are there. So this PDB sum is will summarize and analyze the PDB structure. For the analysis of the PDB structure. Pathways databases are there, like KEG, that is Kyoto Encyclopedia of Genes and Genomes. And these are also some practical sessions. You need to go through them okay, and next is these are some genomic databases, and there are some specialized databases like for corn, the maize database. Tyre is for Arabidopsis. Ensemble, that is a database for human, mouse, and vertebrates. ERIC is erythropathogen and genome databases and national microbial pathogen data resources. JGI is for genomes. JGI genomes, this is for eukaryotes and microbial genomes. MGI is for the mouse genome. So these are the examples. So you need to go through these sites, and you need to develop some understanding about these databases. And these are some other protein sequence databases like UniProt, PIR, SwissProt, ProCite, DIP, PFAM, and protein families. Okay, and Prodom. So these are some other databases for protein sequences, and these are some more databases like for protein data bank, structure classification of proteins. Scope is for protein structure classification. Swiss model, a server and collection of protein structure from PDB acting as templates. So they will work for a template for you. And Modbase is a database of comprehensive structure models of proteins. And these are some other interaction data, protein interaction databases like String, DIP, database of interacting proteins, Bind. So these are actually, and some metabolic pathways databases, BioSic, KEG, Magnet, Reactome. So these are some, okay, these are some microarray expression databases, microarray EBI, NCBI, and University of Manchester, and the database is MAXD, SMD, GPX. This is the Scottish Center for Genomic Technology and Informatics. So these are some microarray expression databases. These are some databases, mathematical databases for like cell ML biomodels. So these are some mathematical model databases. And these are some meta databases, like the data type of database source or platform hosting different database sources. They will host different databases, presenting the data of these databases in new and rather simpler unified form, like entries, EU genes, EU cards, source, and so on, and bioinformatic harvester, etc. And that's it. Am I clear?